Hello students, welcome back to the channel. In this video, we are going to study how the cell control enzyme activity. Now see one thing, enzyme regulate chemistry of cell. But question is, who is controlling the enzyme? One method of controlling the enzyme is we should stop its production if it is not required, or we should allow its production when it is required. Means we are controlling the enzyme production at a gene level. Second question is, if suppose enzyme is produced, then how we will regulate it? Once the enzyme is made, how we will regulate it? Then we are finding two different methods are there. One is they are actually producing allosteric enzymes. Okay, they are producing allosteric enzyme. So here I've just given you this map to understand how the regulation of enzyme mediated reactions are there. One is by controlling substrate concentration or concentration of enzyme, means the enzyme production. Okay, beta. And second is if suppose enzyme is produced, how we will regulate its activity. The cell has to regulate it. Now see next. What are allosteric enzymes? Allosteric enzymes are those enzymes which are having, apart from the active site, they are having some other site also. These other sites are known as allosteric site. Now, allosteric site can be again of two types. If suppose regulatory protein binds on the allosteric site to stop the reaction, then that allosteric site is known as allosteric inhibition. That is a site for allosteric inhibition, means any regulatory molecule binding on this site will inhibit the action of the enzyme. Another regulatory protein, if they bind on the allosteric site, they can promote the activity. Then we will say that is allosteric activation. So allosteric enzymes are those enzymes which are having allosteric site where regulatory molecules can bind. Now, these substances which are affecting the enzyme activity, we call them as they are regulators. They may either inhibit or they may either stimulate also. Is it clear? See one diagram. See in this is a allosteric activation. This is a activator molecule. When it is binding on the allosteric site, it is actually making the active site more comfortable for the substrate to bind. See here, this active site is not comfortable for substrate, but once the activator is binding, active site is getting changed so that the substrate can easily bind. So this is allosteric activation is going on. This is allosteric inhibition example, in which regulatory molecule, which is inhibitor in nature, will bind to the allosteric site and active site shape is changing. So as a result, now substrate cannot bind. Please beta understand very carefully. Two type of allosteric regulation is there. One is allosteric activator, one is allosteric inhibition. Is it clear? Generally, allosteric inhibition is also known as non-competitive inhibition. Name is telling inhibition means it will stop the activity. And activator name is telling it will promote the activity. Understood the basic concept, beta? Now see next. Example is this one. Let us assume this is an enzyme with active site and this is the allosteric site which is actually blocked by inhibitor molecule. Inhibitor molecule has made attachment there with the allosteric site so that active site is not able to bind. So the enzyme is inactive. Clear? And the name of the enzyme is protein kinase. Now, if suppose I have to make this enzyme active, then cyclic AMP. Cyclic AMP, what it will do, it will bind with the inhibitor protein and it will make this regulator inhibitor inactive. Is it clear? The cyclic AMP will remove the inhibitor. And once the inhibitor is removed, it means now substrate can properly bind to the active site and enzyme substrate complex is made. So this is a just simple explanation trying to make you understand how allosteric enzymes are working. Clear, beta? Now see next. Another type of allosteric regulation is cooperativity, which we study in the hemoglobin. We know hemoglobin has got 4 polypeptide chain, 2 alpha and 2 beta. And what we find, if suppose one oxygen molecule bind to one site, it increases the affinity for oxygen molecule for the remaining binding site. Means let us assume substrate has done the binding only on one side and it is making the other site also becoming active so that now they can easily accept the substrate. Is it clear? So it is also a type of a allosteric regulation, which we call it as cooperativity, in which they are trying to amplify the response of the enzymes to substrate. Clear, beta? Next, feedback inhibition. If suppose biochemical pathway in which product of one step is working as substrate for another, and again it is going on, it is catalyzed by multi-steps, and at each steps enzymes are there, what will happen? The product will stop the synthesis of this thing because it is in excess. Suppose the product is in excess, then what will happen? It is a type of genetic regulation in which product will inhibit the first step only. Is it clear, beta? Now, if you see this is final product is binding to the allosteric site, and as a result, substrate is not able to bind. So feedback inhibition generally we see in the biochemical pathway in which the final product can regulate the enzyme activity by binding on the allosteric site. So I hope so this video might be helpful for you. If you have still any queries, please mention in the comment. I will try to help it out. Thanks for watching.